All right. Good afternoon, Epidemics.
Good afternoon.
I have been hearing from a lot of other faculty members lately and from my other classes that stress levels have been going up. So I thought I would kind of casually survey you guys. I see some of you are doing pretty good. Oneida thinks she's a 9, which that's a cute possum right there. That's a happy, cute possum. Personally, I'm feeling the stress from all of the world events and the fact that we're in the second half of the semester, which, well, pretty much any time during the semester is stressful, but yeah. All right. So we do have a quiz and journal entry due Sunday before midnight, as usual. The projects are going to be due on Sunday, May 1st before midnight. If you are going to do a presentation, those are going to be April 26th and 28th. I still have a few students who have not signed up, most of whom are not here today. Julia, go ahead.
So I'm doing a presentation. Did you say something last class that you're going to email the kids who are doing presentations, like what they should focus on or something? Just like,
Yes. Yes.
I don't want to make it like too short, obviously, but I just want to know exactly like what you're looking for, like a rubric, I guess. I don't know.
Yep. I'm going to be sending out those instructions, and the plan is that's going out today. Okay. And then you can, if you have a preference for Tuesday versus Thursday, you can let me know by email in response to that email.
Okay.
I was just looking this morning to see how many we've got, and it looks like we'll be able to get everybody in on those 2 dates.
All right. Thank you.
You're welcome. Any other questions about the project or the presentations?
I don't know if you already said this, so sorry if you did, but so I'm writing like the fiction story. Would I be presenting that?
No, that's going to just be submitted either on Blackboard or by email to me.
Okay, perfect. Thank you.
I look forward to that. Okay, so we are hip deep in the plague. Last time when we wrapped up, we were talking about Yersinia pestis and how it's a bacteria.
I still don't have an appetite from last week's lecture. That was so gross.
Well, this week is not going to be so biologically gross. All right, so bacteria. These are living, single-celled organisms. They have small, simple cells. And nowadays, we can treat them with antibiotics. Antibiotics only work on bacteria. They will not work on a virus. So if you have a viral infection like COVID, antibiotics do nothing for you. Antibiotics were first developed in 1909, so the early part of the 1900s. Before this time, in wars, more soldiers died of disease than from enemy action. So before we had antibiotics, the biggest killer of soldiers during the war was bacterial infections. You could get a cut on your hand, it could get infected, and that could be enough to kill you. So we don't think anything about getting small cuts. You know, you get cuts on your hand, or at least I get cuts on my hand all the time. I'm an avid gardener, so I'm always outside and I don't always wear my gloves. But 120 years ago, that could have killed me. So antibiotics work in a couple of ways. I'm going to list you the most common ways that antibiotics work. They attack the cell wall of bacteria. So our cells do not have a cell wall. But bacteria, if there's your little bacterial cell, outside of that is another layer, a cell wall. This is a tougher structure than a cell membrane. It helps protect the bacteria from harsh environmental conditions, as well as from attack by things like the immune system. Because our cells do not have a cell wall, antibiotics that attack the cell wall tend not to have a lot of side effects, or as many side effects. Others stop reproduction of bacteria. So you've already got the bacteria in you, but it can't reproduce. This gives your immune system time to come in and kill those bacteria before they get too much of a problem. And they can stop protein synthesis or manufacturing. So they stop the bacteria from being able to make proteins. These last two are more likely to cause side effects in people. And that's for a couple of reasons. So first off, you have a microbiome. We mentioned this last time. Your microbiome is made up of a lot of different bacteria, and it's very, very important for a lot of your normal bodily functions, especially in your digestive system. So an antibiotic that's attacking a bacteria is also going to affect your microbiome. Which is why it's very common to have diarrhea, nausea, stomach cramps, and an increased risk of yeast or fungal infections. So these yeast and fungal infections, just like the bacteria, we have yeast, which is a type of fungus, living on our skin at all times. And normally the yeast and the bacteria have a balance. The bacteria keep the yeast populations in check. The yeast otherwise would overgrow. They would become too populous. They would be too much of them on your skin, and they can cause problems. In women, we think of yeast infections. Men, jock itch, and both men and women, things like athlete's foot. So there are multiple yeast or fungal infections that they're all caused by the same fungus. It's just where they happen. It's very common to get these yeast or fungal infections after taking antibiotics. Okay, so that is, oh, geez, I'm out of space. So take a screenshot so I can make some more space here. I'm just going to erase sections. Ah, I didn't mean to do that last one. Okay. We were talking about the why of side effects, and we mentioned the microbiome. There are things that you can do to reduce the risk of yeast infections when you're on antibiotics. There are various kinds of things you can get at the drugstore that are supposed to contain bacteria. It's a mixed bag how good those are, but anything like yogurt or fermented foods, like kimchi, those kinds of things will help you reduce your risk of getting a yeast infection while you're on antibiotics. So you want to take your antibiotics and then an hour or so later have some of this kind of food to kind of help repopulate the bacteria in your digestive tract. You want to do it after the antibiotic has started moving through your digestive system, so that you don't kill off the bacteria in the yeast or the kimchi by taking the antibiotic afterwards. The second reason we have side effects is because of your mitochondria. What do you guys know about the mitochondria?
Powerhouse of the cell.
Yes, powerhouse of the cell. That's what everybody knows. Do you know anything else about the mitochondria?
Produces ATP.
Yep.
Yeah, it transports energy and respiration process.
Yep. So it's very involved in energy production. Where did it come from? Nobody knows. Okay. All right.
Evolved from bacteria, like they have similar structures and there wasn't there.
Exactly. It evolved from bacteria, which means that antibiotics can impact the mitochondria, which of course is going to make you feel crappy, because if your mitochondria aren't working, you're not getting enough energy in your cells. You're going to have fatigue. You might feel kind of brain foggy. There's a lot of things that can go on there. Most antibiotics are relatively mild, have a mild impact on the mitochondria, because if it had a severe impact, that would have a severe impact on our health. Okay. Screenshot if you need it. I'm going to move on to the next one. Now, when we talk about antibiotics, we have to talk about resistance to those antibiotics. So bacteria that are exposed to antibiotics can become resistant or able to survive despite the antibiotic. This is a serious problem. It is increasing, becoming much more common, and it can spread from one bacteria to another, even if they are different species. I'm going to explain how that works in a second. So let's look at this image. We've got a lot of bacteria. We've got a couple of them that are slightly resistant to antibiotics. You take antibiotics, you start feeling better, you haven't finished your course of treatment, but you don't want to take them anymore because you've got stomach problems, so you quit taking them. These bacteria that were resistant now have better conditions. They're not competing with the vulnerable bacteria for resources in your body. And so the resistant bacteria grow and grow and grow. Now, then you take another antibiotic and you do the same thing, and guess what? Then you have multi-antibiotic resistant diseases or bacteria. MRSA is an example of a bacteria that is resistant to multiple antibiotics. And it's a nasty, nasty infection. It's very hard to treat. Nate, go ahead.
Isn't it essentially a staph infection?
Yes, the S stands for Staphylococcus or staph. Yep. I forget what the M stands for. It's a specific antibiotic. Resistant Staphylococcus aureus, commonly called staph. Yeah. So I have a friend who, she's a biologist. She got injured pretty severely while doing field work. So she got bitten by a squirrel. They have some nasty teeth. They can do a lot of damage. Oh, Nate pulled in the M word for me. Thank you there. Methicillin. Okay. Thank you, Nate. She went to the hospital. She got the wound treated, but it got infected with staph. And the staph infection knocked her down really bad. It spread up her arm. She had to have multiple surgeries. And she ended up losing part of her finger where she got bitten because the bacteria basically killed all the cells there. And now she's got a scar going all the way up to her forearm where they had to go in and remove tissue that was very badly infected and necrotic. It's been over a year. She's still having to get healthcare from it. So antibiotic resistance is not something to joke about. Nate, go ahead.
How do we combat this? Like, because this seems to be a serious issue. And I'm wondering if there are efforts or technologies that have significant advancements into destroying resistant bacteria?
We're right here on my notes. That question will be answered right here. A
I
good
recall-
question.
My dentist being reluctant to administer antibiotics to me too often because of this issue. So, yeah.
Yep, that's part of it. Yes, we're going to cover that in great detail. Good question, Nate. All right. So take a screenshot if you want. I want to explain next how a bacteria, oh, good grief, cat wants attention. How a bacteria can spread antibiotic resistance to other bacteria. So I'm going to do some bad drawings of bacteria. So here is a bacteria. And here is a different bacteria. So let's say this one is Staphylococcus aureus, and we'll call this one E. coli. So Staphylococcus aureus has within it these little circles of genetic material called plasmids. These little circles are not the main part of the genetic information for that bacteria. It's extra genetic material. It's not required by the bacteria. The bacteria can survive without these. Plasmids generally have genes for something that gives the bacteria an advantage, like being able to digest a different food, or in this case, antibiotic resistance, but also things like traits that help it evade the immune system. So lots of different things can be in the genes on this plasmid. And I've drawn one, but the bacteria is going to have multiple copies of it. So the Staph aureus comes along and it spots this other bacteria, and it does something weird. It builds a bridge. So this bridge between the two, this process is called conjugation. And it's the closest bacteria come to sexual reproduction. Because what can happen then is these plasmids can move from the Staph aureus into the E. coli. And now we have E. coli that has, in our example here, antibiotic resistance. These aren't even the same species. This is like you giving your ability to digest milk as an adult to a snake. It's bizarre. Bacteria can pick up DNA from the environment as well. So this Staph aureus could die on the countertop, and that gene will survive for a little while, and another bacteria comes along and can pick up that gene. I mean, there's all kinds of weird ways that genes can be transferred between different kinds of bacteria if they're on a plasmid. Does this make sense to people? It's weird, it's bizarre, but do you understand it?
Yes.
Thank you, Jerry.
It's like Frankenbacteria.
Yes, Frankenbacteria, absolutely. Okay. This commonly happens in hospitals. We know why this is. You can think about this, right? Why is this?
It's a hub of bacteria there.
Petri dish.
Yeah, it is. It is. Hospitals are absolute petri dishes, and a lot of people will be on antibiotics, so you're more likely to get antibiotic resistance showing up. If the healthcare people don't wash their hands well between patients, they can carry this antibiotic resistance. Doctors who wear ties. I hate to see a doctor in a tie, because how often do men wash their ties?
Wow.
Right? And you bend over the patient and the tie falls down, right?
Yep.
They're just great ways to communicate, to spread bacteria between patients.
But I think this benefits us as well, because when we're trying to genetically engineer bacteria to our benefit, this works in our favor.
This is exactly what we take advantage of.
Haha.
Yep. All right, screenshot if you need it. Okay, gonna go to a whiteboard real quick. All right, so Nate was asking just a second ago, what are we doing about it? So I want to talk about antibiotic development. This is a relatively slow process. It is extremely challenging to develop new drugs, regardless of whether we're trying to treat bacterial infections or heart disease or any kind of condition. It's a hard process, and it takes a long time, and it's expensive. So antibiotics in particular are high cost to develop, discover and develop, and low profit margin. There is not much funding from the government for this. And whether we are aware of it or not, government funding of science is critical to developing new medications. So governments fund science primarily at universities. So if you are at a university, you're working on an advanced degree, a master's degree, or a PhD, you can sometimes get funding from the government to help you do research, especially if it's related to medical care. The research that you do, however, is what we call basic research. So when I was in graduate school, I was working on studying a specific molecule in the mitochondria. It's very important in the mitochondria. It's important for body heat regulation. It's important for antioxidant function. So it's got a lot of different functions in the cell. But I was only looking at this one particular molecule. And that is the kind of research that pharmaceutical companies are not going to fund. Why would they fund me looking at this molecule? It may or may not ever be relevant to developing a drug.
Not profitable.
It's not profitable. But if suddenly a new disease comes along that attacks and destroys this particular molecule, now all of a sudden everybody wants to know how to protect this molecule. And so they want to know all about this molecule. But if nobody's been researching it because there's no funding for it, then you've got this, great unknown. Nobody knows anything about it. So, I mean, when we think about our current epidemic, before COVID, there were not a lot of people studying that group of viruses, the coronaviruses, because frankly, most of them cause common colds. We've all had a coronavirus infection that caused a common cold. And who wants to fund research on that? So all of a sudden, when COVID comes along, there's one lab who is the world expert on the topic. And all of a sudden, everybody who's ever worked in that lab, governments are wanting to hire them, pharmaceuticals companies are wanting to hire them. I mean, it's the hottest thing ever. And yet 3 years ago, it was like, why are you studying this? So government funding of science is important. Learning the basics is important. We don't always know when it's going to be relevant. All right, pharmaceutical companies get 5 years of exclusivity when they develop a new drug. So for the first 5 years, nobody else can make a different version of that drug. So there's no competition for the first 5 years. And part of that is because of the expense of drug discovery and development. 5 years may not pay off the development cost. So not all drugs end up being profitable for the pharmaceutical industry. And most drug development projects fail. They never produce a drug. So pharmaceutical companies are a high risk, potentially high reward company. It's a high reward if you happen to get something good. That said, there is pressure on pharmaceutical companies to be profitable, not necessarily to save lives. And we saw this with insulin recently. I think insulin prices are still horrifically high because insulin was no longer being protected. Anybody could make it, but it's expensive to make, relatively speaking. So not very many companies were making insulin. In fact, they're still not. So the couple of companies that were making insulin, they got together and decided to raise prices. This was just pure greed. and they raised the price to the point where a lot of people who needed insulin could not afford it. It went from being a relatively cheap drug to being a very expensive drug. So this is one of the reasons people hate the pharmaceutical industry, because they do this and there's not a lot of regulation from the government. The government doesn't step up and say, okay, we're going to tell you you can't charge this much because of, the freedoms. All right. Take a quick screenshot of this. I've got a little bit more to say on this topic. Today, in the US, around 23,000 people die yearly due to bacterial infections. It's not up there with heart disease, but it's not a small, rare thing for people to die of this. We don't think about it because most of us haven't known anybody who had a severe bacterial infection. But it's true. It's out there. It's a problem. It's a growing problem. The easy to find targets, we've found antibiotics against them. The cell wall, it's easy to find antibiotics that target the cell wall. We found a bunch of those. Now we have to look at some harder targets to attack. So I want to talk a little bit more about the pharmaceutical industry because most people have no knowledge of how it functions. The average cost to develop a new drug is over $2.5 billion. The time it takes from discovering a potential drug market is 6 to 7 years. Only about 12% of potential drugs, once you've found a potential drug, make it to market. Most of them wash out because they have bad side effects, or it turns out they don't work so well in once they get in the body. Jerry, you have your hand up? Go ahead.
Yes, so I've been reading that male birth control is possibly going to be available. So is this why it wasn't already available, these things that you're mentioning now?
Male birth control has been 5 years into the future for at least since the 80s.
Oh, damn.
Side effects are a big part of it, which is ridiculous because female birth control does have some pretty nasty side effects, especially for some women. But the idea is that men will not be willing to deal with the side effects.
Oh.
And since men aren't the ones who get pregnant, the consequences are less severe if there's an unplanned pregnancy.
I get it.
Even though it should be shared equally, it's not.
We're fertile all year round, so we need it more than anybody.
Women are fertile every month of the year for a few days.
Every day, all the time.
Yeah.
I can pop some babies out.
Yep. Yes, you absolutely could.
Hugh Hefner got a woman pregnant at 84. Like this is a lifelong thing. Forever we're baby factory. So get that going, please.
Yeah.
What did I
It's called
just
a
come back to?
vasectomy, Jerry.
Male birth control. Would you say, Professor?
It's called a vasectomy.
Right, but that's a whole different deal. You know, some are reversible, some are, I can get off of birth control and then have a baby.
That's true.
So
That's
funny
true.
saying this.
Yeah, I'm down. I'm down for male birth control.
Yes.
But
What?
with vasectomies, I know if you try to reverse it, there's a chance that you can't get like that ability back. If you know what
Exactly.
I mean. Yeah, give me a shot, give me a pill. Yeah, I'll take it.
Yeah. But the pharmaceutical companies think that you guys would not be willing to do it.
Let's start a petition. Let them know we need 100,000 signatures.
Get that on change.org, Jerry, right now.
And as I mentioned a second ago, you need a target. So one of the problems with, for example, developing treatments for Alzheimer's is we don't know what causes Alzheimer's. We don't know what the cause of that disease is. We know things that contribute to it, but until we find a molecule that we can go after, we're just kind of like, you've heard the phrase shooting fish in a barrel for something that's easy. Well, it's kind of like shooting a shotgun into the ocean and hoping you catch something.
Needle in a haystack, I think would be another good one.
Needle in a haystack, absolutely. All right. So what can we do to deal with antibiotic resistance? What can be done? There are some things that you personally can do and some things that could be done at the government level. So at the personal level, don't buy products with embedded antibiotics. So a few years ago, they were literally selling things like kitchen countertops that had been embedded with antibiotics to keep your kitchen sterile. Well, that's just going to lead to antibiotic resistance in your kitchen. Hand soaps. Yeah, Nate, go ahead.
Sorry, just to add to that. So this is kind of like a group effort thing. Like we all have to contribute to fighting this. It isn't just something scientists do, right?
Right. Right. Science can provide the how to deal with something, but there needs to be the social side as well, because I can tell you what we could do to deal with climate change, to get rid of COVID, but on a practical basis, I don't have those answers. And changing public opinion, I don't have those answers. Those are answers that we need from what are called the social sciences, psychology, sociology, and that sort of stuff. And that's not something that I was ever trained in. All right. Your hand soap, guess what? Soap and water kills 99% of germs on your hands. You don't need antibiotics in it. Soap and water does the job.
Does the water have to be hot? Because I've seen people like talking about this back and forth, back and forth.
No, water does not have to be hot. It's a physical process. So temperature does not matter. The only way that temperature matters if you're talking about washing your hands is if the water's too cold, you may not wash your hands long enough. Although if you get something really disgusting smelling on your hands, you want to wash in cold, because hot water opens up your pores and the smell can get into your hands before the soap can get the smell off. You don't want to know why I know that. Okay. Only take antibiotics when and as prescribed. And if you've got a doctor or a dentist prescribing antibiotics, ask the doctor why. Is this an antibiotic infection? Am I likely to get a bacterial infection? You know, with dental treatments, some people are at risk of getting various kinds of infections, especially with more severe dental work, more intense dental work. Some people are just not very susceptible to that. So you always want to question that, because they're the old school doctors, and they're not always old doctors, believe in antibiotics as a preventative, and they're not always needed. At the government level, limit healthcare costs, especially drugs. People won't take antibiotics if they can't afford them. And there are a lot of people who have to make a decision. Do I eat or do I buy this prescription? That is not okay. Most developed countries have good government healthcare. We basically don't. We're the oddball on that one. The government can also limit profit, maybe as a percentage of sales, or based on how much money a company puts into research. Maybe allow more profit on medications that are riskier to develop. Maybe there's not a big population pool. In fact, the US government does provide support for companies that go after rarer diseases.
Is that in the form of subsidization?
Sometimes it is, sometimes it's tax cuts, which are basically subsidies as well. Yeah. And of course, the government needs to fund that basic research. Even if we don't know why it might be relevant. All right. So that was my big spiel on antibiotic resistance, the drug industry, or the pharmaceutical industry. Nate, yes.
This is kind of related to the research, but it's kind of like a morality thing. Do you think it's possible to completely like stop testing on animals completely for pharmaceutical advancement? Okay.
Not yet. I would like to see that happen in my lifetime, but we're going to need some major technological advances before we get there. Right now, we know of thousands of different kinds of cells in the human body. We're still finding new types of cells. And a lot of these cells cannot grow in tissue culture. You can't grow them in a lab. So finding a way to test a drug on every type of cell that we have, it's basically impossible right now. And we have to test it on every type of cell in the body, because what's toxic to the liver might be perfectly fine if you test it against kidney cells. What's toxic to the brain might be fine if you test it against liver cells. So that there's that part of it. And there's also the fact that sometimes drugs are changed primarily by the liver. So the liver tries to break down toxins in the body. That's one of its jobs. It purifies the blood.
Is that metabolization or is that a different process? Okay.
Yeah, it metabolizes them. That's one way of putting it, yes. So it changes these potential, what it thinks are toxins. Sometimes the byproduct of that process is more toxic or more effective as a drug. But we don't know that unless we run it through a functioning liver. So, okay, to some degree, we don't have complete functioning liver in tissue culture, but we can test on liver cells. But if the liver cell converts that drug to something that's toxic to nervous tissue, well, then we've got to run the drug through a liver and see what kind of effect it has on the thousands of different kinds of cells. So unfortunately, animal testing is still necessary. And there are lots of drugs that look great in cell culture and then don't do anything when you put them in an animal, or show up as toxic when you get them in the animal. So yeah.
I appreciate your insight on that. Thank you.
You're welcome. One of my first jobs, I worked at a pharmaceutical company, and we were working on treatment for human papillomavirus, which the company never got a drug for human papillomavirus. And now we have a vaccine, thank goodness. We were trying a topical cream that we put on the skin of mice. And the first test was looking at 5 mice. We put the cream on the skin of the first mouse. By the time we got to the 5th mouse, the first mouse was dead. It didn't kill cells in tissue culture. We still don't know how it got into the body and killed them that fast, because that is incredible. Just think about that. Something you put on your skin that in like 15 minutes it kills you.
And this was meant to prevent the virus, this cream.
It was meant to treat the virus. Yeah. It treated the virus great in tissue culture. But that convinced me that animal testing is still quite necessary, because it looked good until we put it in the mice.
I guess the morality thing comes in when we're dealing with domestic animals, like dogs, or that's like what bothers me, like testing on like dogs, cats, monkeys.
Yeah.
Things of I don't want to say sentience, but you get what I'm going with, right?
Yes.
Yeah.
Yes. Yeah. I definitely hear you, and I definitely feel that. I agree with you. I don't like animal testing in general, and certainly animals that are more like us or more familiar to us bothers us more. Unfortunately, mice and rats are not always the best animal model for drugs. And so we've developed, we do testing in other animals that we know, I'm trying to remember which specifically, but I know, for example, that diseases that treat the heart don't always, we can do toxicity testing in mice, but for how effective it is, we need to work in, generally speaking, we work in dogs because their hearts are more like ours than a rat's heart or a mouse's heart. So unfortunately, that's why we need to use the different species. The only alternative is testing untested drugs on people, and that's even more morally fraught. It's a complicated topic, and like a lot of complicated topics, there's not a clear, easy answer. Whenever you start talking about morals and ethics, a lot of times, I mean, sometimes there's always an easy answer. Should you punch Nazis? Absolutely, always, no problem. Testing on animals, oh, now it gets kind of
It's
difficult.
not black and white.
It's not black and white.
This reminds me of when they were bringing the cure of smallpox to South America, as you told us, and they had the orphan boys.
Yes. Yep. It's a tricky thing. All right, I'm going to go back to the slideshow. I don't think I've added anything while we've been talking about ethics.
I just wanted to add one more thing. Speaking of Nazis, isn't it true that we got a lot of scientific knowledge from like the gruesome testing and other conductions that they did, or is that like exaggerated? Because I hear that sometimes.
Well, we took this first off. I know that.
Yeah,
We took-
Operation Paperclip, it was called.
I don't think we got as much from the Nazis as the modern day Nazi sympathizers want you to believe. Because a lot of their experiments were just, they were not scientifically done and documented. I mean, if you're doing stuff like this, you're not documenting it. And that means nobody's going to be able to go back and see what you did.
Yeah, and there definitely was atrocities when you talk about Joseph Mengele, the angel of death, and all the stuff that went on.
Oh God, he was terrible
Yeah,
with the
twin
twin
boys.
boys.
Yep.
Yeah. Yeah. All right. So now we've talked a lot about biology the past couple of lectures. Now we're going to talk a little bit more about the social aspects of the Black Death. So a lot of the information here, we're talking about the second, plague pandemic. So a lot of this information is not just limited to the Black Death. The Black Death refers to the first few years. The second plague pandemic was much, much longer than that, and it ran from the 1330s to the mid 1800s. So you think COVID's bad? You think it's been lasting a long time? Oh, okay. So what we know that approximately half the European population died, mostly during the Black Death years. We know that around 1/3 of the Middle Eastern population died, again, during those first 5 years. We know the population of China declined dramatically. We don't have good numbers from there, but we know that the population there around 1200 was an estimated 120 million. And in 1395, the population was around 65 million. So that's almost half. But this was, 130 years before the Black Death came along. We talked about all the environmental changes that were going on right before the Black Death. So we can't say that that entire decline was due to plague, but obviously there was a significant plague. During the same time, there was a massive famine and a lot of political upheaval. We know the Black Death went to Africa. We have no information about population trends during this time period. And we don't have this information for two reasons. One, fewer scholars have studied this time period. And colonialism destroyed a lot of traces and documents from this era. Well, basically from any time before the Europeans arrived. So the European story, of course, Africa was a primitive nation. It's like, oh, yeah, no, it wasn't. There were, empires and cities and advanced civilizations in Africa before the Europeans came in. The Europeans just wiped them out and wiped out as much evidence of that as possible. Okay, good time for a screenshot here. So, what were people thinking was going on? Remember, the whole second plague pandemic is before we understood about germs. We didn't know what bacteria were at this time point. So there were a couple of major concepts for what causes disease. We're going to talk about these two, although both the imbalance of humors and miasma were very much linked together in people's minds. So bad air could imbalance your humors. So these are, Nate, go ahead.
You just elaborate humors. What is that? Because I hear humor and I think funny, ha ha ha. But what is that in that term? Like, what does that mean exactly?
Yes, I'm going to explain it. Yes, perfect question, perfect timing. This is a graphic that shows the four humors. So the four humors were blood, yellow bile, black bile, and phlegm. So basically, we're really looking at things that we can recognize. We recognize blood. We know what that is. Phlegm, that's mucus. Okay, so we know what that is. Yellow bile and black bile, that's going to come from vomiting. people would vomit and it might look kind of yellow-green or it might look kind of black. And so those were believed to be because you were expelling yellow bile or black bile from your body. Each one of these humors had characteristics according to the ideas back then. So blood was associated with air, and it was considered to be hot and wet. Phlegm was associated with water, and it was thought to be wet and cold. Black bile was associated with the earth, and it was cold and dry. Yellow bile was associated with fire, and it was hot and dry. What did these terms mean? Well, honestly, not a whole heck of a lot, because this is all, we've proven it. We know this isn't true anymore. So I want to go through these real quick. So I'm going to have to go to the whiteboard for this. All right, so blood, hot and dry. And this was associated with enthusiasm, activity levels, how social you are. People who were attractive with pretty skin had high levels of blood, or at least that's what they said. Yellow bile. Oop. Hang on. Blood was wet, wasn't it? Let me just erase this because I can't remember those off the top of my head. So blood, enthusiasm, active, social, attractive with good skin. Yellow bile is associated with aggression, short temper, daring or adventurous, and of course, yellow skin. Yellow skin nowadays, we know that that's a sign that you're having liver problems. Jaundice.
Yeah, jaundice. Yeah.
Black bile was associated with depression, laziness, fear, and being sickly. And it could be seen in people with black hair, black eyes. Phlegm is associated with apathy, tumors, rheumatism, which is a type of arthritis, low spirits, forgetfulness, and white hair. So apparently when you get old, you have an excess of phlegm. So it was very important to maintain a balance of humors. And how do you do that? Avoid displays of emotion, and drink sparingly. Avoid excess exercise or sexual exertion. Not too much sex, folks. You're going to knock your humors out of balance. And guard against sudden chills and drafts. So, does this sound like anything we hear about today?
In the South, my grandma might say some weird stuff like that.
Have you ever seen a commercial that talks about keeping things in balance? And they're trying to sell you like a fancy yogurt?
Even the alkaline water and all that stuff. But
The
yes.
alkaline water stuff? Yep.
I know what you're talking about, but I forget the name of the yogurt. I'm trying to think of it.
Activia.
Yeah, that's it. Yeah. Now, I have a question too in regards to cold weather. Can you actually become ill? I'm not talking about hypothermia, but they're like, oh, put a coat on, you'll catch a cold. That's a myth, right?
That is a myth.
Okay.
Yes. Hypothermia, of course, is one thing, but just being cold or going outside when it's cold does not cause disease. We know that some diseases are more common in the winter. Part of this is probably due to spending more time inside and interacting with people indoors a lot. So you're sharing your cold viruses. But we don't fully understand why certain diseases happen at certain times of the year. And it's very, very, very distinct. If you go in with a head cold at a certain time of the year, the doctors will know it's probably rhinovirus versus one of the common cold coronaviruses. Those happen at different times. Nate, go ahead.
But there's no link between like cold exposure making you predisposition to the common cold or rhinovirus, and they don't extenuate factors that could lead you to more probable infection. There's no link like that.
Not for the average person going outdoors in the winter for an average length of time. It's like you said, if you're out for a long periods of time, then having to deal the physiological responses to dealing with being out in very cold weather for very long time periods can impact your immune function a little bit. But when I say extreme, I'm talking, spending a week camping in the snow. Oh, good grief. Okay. And it is time for class to end. Any questions on this? When we come back, we're going to talk about how plague was considered an excess of blood and what people did to try and treat it. And we'll talk about miasma.
I have another health related question.
Okay.
Of course, it just slipped my mind. Yep, nope, I lost it. Sorry.
Well, you can always email me.
See you guys next week.
Bye. Next week.
Wait, then we have class Thursday?
Yes, we do.
Okay, yeah, I'll email them. Keep them up to speed. Bye-bye.
Bye.
Have a good day.
You too.